Welcome, everyone. My name is Andreas. I'm the Communication Manager for the Schaar Lab, and I'm very happy to invite you all to our session, Revolutionizing Healthcare. Today, we will talk about how to use machine learning to power clinical trials. We have some very exciting guests for our roundtable. When we start, first, talking about what this session is all about in its general outlook. We try to build a shared community and develop a common language when it comes to AI and machine learning for healthcare. And I hope these sessions will in the future grow this community. I think in the past, we already had some very interesting discussions and topics that we talked about. For example, our last two sessions were about synthetic data, and we also had some leading clinical voices share their visions about AI and machine learning. Today, we will start with a short introduction from Mihaela in a minute, and then have some introductory presentations from our guests who will frame the situation of machine learning and clinical trials, where are we now and where we might go. And for this topic, we will go deeper into doing our roundtable when Mihaela is asking some very specific and interesting questions to our panel. You have the chance at the end in a Q&A to ask all your questions. If questions coming up during the presentations and the roundtable discussion, please feel free to write them in the chat, and I will either read the questions or we will ask you to pose your questions in person at the end of the presentation. We should wrap up around 5:10. And now I'm very excited to go to Mihaela, and Mihaela will introduce the topic of today. Thank you very much.
So thank you very much, Andreas, and welcome everyone that is joining us. And especially I'd like to thank our 4 panelists today, which Andreas will introduce soon. We have talked over the past months about a variety of topics where machine learning can really make a difference in healthcare. But at the core of adopting, whether it's new interventions, whether new practices or new therapeutics, is the way in which we conduct clinical trials. Because in the end, many of these interventions that we are discussing cannot become practice and they cannot become used unless they are validated by clinical trials. Yet clinical trials are still very much the same, even after all these many years that they have come into existence. And the question is whether now, with the data that becomes available and the technologies that become available, whether it's in statistics or machine learning, we can go and empower this golden standard of adopting interventions, that is clinical trials, whether we can improve them, whether we can empower them and bring new ideas to transform them, thereby bringing useful technologies to patients and clinicians in a more rapid way and a more effective way. But this is a complex topic, so I'm very fortunate to be joined today by 4 panelists who have different pieces of the puzzle. They have all been involved in clinical trials over the past years and at times decades. And each one of them has been looking at clinical trials from a different perspective. They are all clinicians, but their perspective on clinical trials has been very different. So I'm very fortunate that they all 4 accepted and provide their unique insight on that. We, on the other hand, from a machine learning point of view, we have been starting to put together small pieces of the puzzle. You can see on the link highlighted here by Andreas, a few of our very preliminary works in this area. But I hope that together with the entire community in Revolutionizing Healthcare, we can go further together and understand what problems machine learning can solve. And Andreas has not mentioned that yet, but actually we are proposing a double header. In the first session today, we will discuss ideas and how can clinical trials be potentially improved. And then in the next session, we are going in July to look back at this session today and together with the 4 panelists, try to propose some ideas about what machine learning can do to indeed improve clinical trials. Thank you very much again for joining us.
Thank you very much. Yes, indeed. So if today we can catch your interest for this topic, there will be the next one coming in July. Looking forward to that very much. And now I'm very happy to be able to introduce today's panelists. We have Professor Carsten Utoft Niemann, who's the Associate Professor and Head of the Chronic Lymphocytic Leukemia Laboratory and Clinical Research Program at Rigshospitalets Department of Dermatology in Copenhagen. And from where he's joining us today, we have Dr. Eoin McKinney, who is a university lecturer in renal medicine at the University of Cambridge and a honorary consultant in nephrology and transplantation at the Cambridge University NHS Foundation Trust. We have Dr. Pierre Marquet, who is the head of the Pharmacology, Toxicology, and Pharmacovigilance Department at the University Hospital of Limoges, and president of the European Association of Clinical Pharmacology and Therapeutics. Welcome to you as well. And Professor Richard Peck, who is the honorary professor in pharmacology and therapeutics at the University of Liverpool and former global head of clinical pharmacology at Roche. So there are a lot of interesting perspectives to come. We will have those short introductory talks now. There will be roughly 3 minutes, and they set the introduction for the discussion later. Machine learning for clinical trials, where are we now? We will start with Dr. Eoin McKinney, who I will invite now to take the word. If you would like me to change a slide of panelists, just let me know and I will press forward. Thank you very much, Eoin.
Many thanks, Andreas. I only have 1 slide, so you shouldn't need to be forwarding anything. But I do have it there, if you could forward onto the slide. Thanks very much. So this is an attempt to frame the subsequent discussion that we're going to have. And in the center, you can see the conventional approach to clinical trial design, where we start with the preclinical work going through the conventional phases up to approval, post-market approval, and subsequent surveillance. And the amount of data that we accumulate as we go through that process about a given drug or about that given intervention that's being trialed increases. So you start with less, you go on and get more. But of course, the amount of information that you may have from previous trials or about the disease context in which you're trying to make your intervention can be substantial. And it's also worth pointing out that the chances of successful completion of each of those phases is actually quite low. It varies by therapeutic area, but it's 3 to 10%, a bit higher for vaccines, but actually it's pretty poor, and there's substantial scope to improve that process. And the question becomes, how might we do that? And the idea is that there's an increasing amount of data that we might be able to tap into. So historical trial information may not be available, but there are increasing ways in which federated access platforms can give us access to historical trial results that can be used for learning. There's a lot of similar collated data on individual drugs and on individual diseases, and there's increasing ways of accessing those to better understand the context in which a trial is done. Obviously, there's collation of data, clinical data through electronic health records, trial monitoring data, and increasing wearable technologies that can provide a wealth of data on clinical trial subjects. And that can be deployed either for trial design, actual monitoring during the trial itself to improve monitoring or collation and collection of data, and also afterwards during analysis. In terms of methodology, which is the second tier down, there are the methods are advancing rapidly in terms of what might be deployed on that increasing amount of available data, from causal inference that might allow us to identify with individual effect estimation what interventions might be useful before we even go on and test them in the context of a trial, through to prediction or identification of markers or surrogate endpoints that we might want to measure in the trial as an endpoint rather than waiting for a full clinical readout. And these can all feature at the stage of trial design, or monitoring in the middle, or analysis afterwards, or we could discuss afterwards. I think what that results is in huge potential for machine learning to change the way in which trials are being done. I think it's fair to say that that hasn't really been done yet, not systematically, and there's a lot of potential, but it hasn't really been realized. So I think the discussion will be very interesting as to how we might make that, how that might be possible, because the benefits are huge. Trials could be shorter, they could be cheaper, they could more successful. And in fact, they could be self-sustaining or improving in a way that when I do a trial now, that might improve my ability to do a related or even a different trial next time round. The challenges here that are outlined are just some of the obstacles in that. And I'm sure that will be a focus of our subsequent discussion. That's more than 3 minutes, so I'll pass the baton on to our next speaker. Thank you.
Thank you, Andreas. Can you show us the next slide, please? So I'm a clinical pharmacologist, so I have a pharmacological perspective on clinical trials, but it's very consistent with what Eoin has just presented. So we, as clinical pharmacologists, we are more interested in a quantitative assessment of efficacy, let's say the effect size, rather than the p-value, the statistical significance of the results. Of course, it has to be significant, but we want to know what is the actual effect size. Also, we are more interested in the benefit-risk balance rather than the way the results are usually presented with clinical trials, where you have a primary outcome, which is generally efficacy, and then you have adverse effects, which are considered as secondary and not really quantitatively assessed. Also, we are keen on hard outcomes, such as myocardial infarction, stroke, or death, rather than surrogate markers or measures such as blood pressure, but maybe we can have better surrogates. And also, we are concerned about extrapolation of clinical trials to real-world population, and we believe that drug effects and benefit risk should be reassessed in this target population. In the real-world population. In the past, several shortcomings have been identified, such as multiple biases in effect size assessment, particularly when using real-world data as opposed to controlled clinical trials. No clear way of combining quantitative benefits and quantitative risks. We all talk about benefit-risk balance, but actually we don't know how to balance them. Also, hard outcomes are too rare and too distant generally, so we need surrogates. We are not happy with the surrogates we have, but we would need better surrogates of efficacy, but not only that, of risk, which means benefit-risk surrogates. We have used a very rough methodology to reanalyze existing data in the past, which is called meta-analysis. And still, it helped identify methodological biases and trying to correct some. And so my question would be, can AI help us to go any further in this? Next slide. So this is taken from one of my colleagues who can do to try and identify biases or shortcomings in clinical trials, such as the influence of sample size, whether the study was a single center or multi-center, whether it was academic or industry funded, the quality of the surrogate markers, whether it was a single arm study or a control study, whether randomization was made individually, patient per patient, or per cluster of patients, and whether it was a randomized or not randomized trial, and if not randomized, what kind of correction was applied, such as using a propensity score, for instance. So I don't expect that AI can solve all of these problems, but I would say probably it would help provide better surrogates. It would probably help correcting non-randomized trials results into something that would look like randomized, and probably also single arm versus control studies. Next slide. Just to show you an example, quite an old one from the same colleague, showing that when you compare double-blind clinical trials with open, can you click again, please? With open studies, you find a huge difference. Those trials were all controlled. The only difference is that in the upper part, you can see double-blind trials where neither the patient nor the doctor knows what the patient is taking as drug or placebo. And in the other one, the open rectangles, the patients and the doctors knew what the patient was taking. And the difference in size is 34% in this example. Next slide. And it was confirmed in many other studies and meta-analyses, so much so that the community has proposed to correct the effect size by an average of 15%. So the community has proposed to correct by 15% the effect size in non-randomized trials, but it's obviously something average, which might induce other uncertainties or biases. So maybe we could come up with something better than this, and maybe AI could help with this. I'm done. Thank you.
And thank you so much. And if you go to the next slide, I'll just give a very brief background on me being a clinician walking between the wet lab and the data lab and the clinical setting, collaborating with a number of pharmaceutical companies and international study groups to develop clinical trials. So that's my background. And if you go to the next slide, you will see my take on where I want to bring machine learning into clinical trials. The green line here represents the standard treatment, and it's just like progression-free survival over time. And we have two different experimental treatments, the blue and the red one. And if we look at this setting, what is my question from the clinical perspective is how to identify the yellow part of the graph between the lines here. That would be the patients benefiting from one of the experimental treatments. And is it actually in just these two dimensions, or would the two experimental arms actually benefit different proportions of patients? I cannot tell this from this regular clinical trials, randomized clinical trials. So if you go to the next slide, what I want us to aim for is to use all the available data on clinical training in the upper part, develop the algorithms, and test prospectively in clinical trials whether these algorithms can actually help us identify the patients who would benefit from a specific treatment in a clinical trial and who would not benefit from it. And obviously, in the optimal setting, define the new treatment that we should apply to those subgroups not benefiting from the experimental treatment here, and develop new translational studies in the lower left part. So my question would be, have we actually succeeded to do this in any trials at the moment? And discussing this with my co-panelists here and looking through the literature, yes, there might be a few examples in like defining dosing of a specific drug, in identifying specific lessons. It could be in images that we can use to guide our treatment, or experiments or clinical trials where we try to use the genetic or the pathological staging to assign different treatment options to patients. But looking through the list of a little more than 100 artificial intelligence models approved by the FDA, just one of those were actually tested in a prospective clinical randomized trial. Thus, we have lengths to do, and that's really what I aim for today, together with the other panelists, to define this pathway, how to implement and how to develop machine learning to identify these patients who would benefit from a specific treatment. Thank you.
So thank you. My area of interest actually builds very nicely on what Carsten's just talked about and generalizes it. As you heard in the introduction, my background is pharmaceutical industry work. I've spent 30 years as a clinical pharmacologist working in the industry. And as you also heard from, I think it was Eoin at the beginning, the success rate in clinical development is low. And what I'm thinking about here is ideas that have come based on experience in drug development, but I think they're equally true when thinking about looking at alternative indications for existing drugs and positioning things from a clinical academic perspective. And that fundamentally is that very high failure rate. And the biggest single reason for drugs to fail in development is they don't work. At least they don't work in the population they're looked at. Genentech, who first developed Herceptin, the HER2 monoclonal antibody in breast cancer, did a very nice theoretical exercise very clearly. If they had developed the drug in an unselected population, they would have needed at least 10,000 patients to show a benefit in metastatic breast cancer, and they would have probably found about a 1-month survival benefit, which nobody would have thought was clinically meaningful. So those have missed the effect of the drug. And we begin to know about this in cancer because we begin to understand that similar symptoms, similar histopathology may have very different molecular bases. And you begin to target the drug specifically to the molecular pathology. But we're probably not doing that in any other diseases. So in how many other diseases might we be failing to find effects of effective drugs? Now, what happens in industry, at least when you do your phase 2 trial and it doesn't work, is you start torturing the data, trying to do lots of retrospective analyses in a desperate attempt to find a subgroup in whom the drug worked and in whom you can then try to keep your molecule alive. I've never seen one of those work. But I still have this problem that I suspect there are some drugs that did work in some patients. In case you think this is purely theoretical, there's a drug called mepolizumab, which is an effective treatment for some patients with asthma that was first developed in the 1990s. I was working in SmithKline Beecham at the time, and it was terminated after a negative phase 2. But it does work if you look in a certain subset of patients. And that was then rediscovered about 15 years later with the result that it is now a drug available for that subset of patients. More recently, the anti-IL-6 monoclonal antibodies were tested in COVID. The initial trials, which were well done, because 2 of them were done by the company I was working for, and 2 of them by another pharma company, but they were well controlled, well designed, rigorously done trials, conclusively showed no benefit in these severe COVID patients. And then REMAP-CAP and recovery about a year later also pretty convincingly showed there is a benefit. The difference was the patients are now treated with steroids. And the anti-IL-6 monoclonals only seem to work in the patients receiving steroids. So they do work, but you have to look in the right place. So I hope that there is a way, perhaps building on some of the things that Carsten talked about, that we will be able to use AI more broadly to begin to understand where should we be looking, and or even where is the signal starting to appear. I'm out of time. I've talked here about finding the right responder, but I think there's also an interest in finding the right dose, which is something industry at least does extremely badly, and where there's considerable room for benefit. But maybe we'll get to that when we get into the wider discussion. We'll come back to that, but I'll stop now.
Now we go over to the roundtable discussion. So our first question, and thankfully Eoin and Carsten have volunteered to answer this one. How can we conduct more effective clinical trials? And Eoin, if you want to start.
Sure, no problem. With respect to for me, a more effective trial is a trial that's cheaper, faster, more likely to demonstrate a clinical benefit. And this follows on directly from what Richard was saying. I think it is a fatal misconception to think that the diseases that we typically try and treat are monolithic ways. Diseases are a heterogeneous mix of different mechanisms and different stages, and it's highly likely that a single intervention is going to affect those people differently. So I think being able to stratify and make our treatment more individual, and by that I mean whether we're selecting which people should be treated, at which time point they should be treated, or with which dose they should be treated. Any of those factors could produce individualized therapy. I guess the big challenge for machine learning is how we know that before we've done the intervention. Because effectively, if a drug has not been used before, then we have no prior data to learn from. But that isn't quite true, because actually we've got prior data on the disease where other interventions have been used, and we may have prior information on similar interventions or analogous interventions, whether they're in that disease or in another context. So I think there are many ways in which we can or could improve the way that we make a given clinical trial more effective. I think some of the challenges for doing that include getting regulatory acceptance for a methodology that is different to what is conventionally done. And what I expect the future to look like is that there will be a creep where we realize that by aiming for these more effective trials, we can have a higher efficacy rate. Once that gets ingrained and people realize it can improve and give us more effective trials, that should snowball. And in fact, if you take a simple example, you look at the success rates of clinical trials from phase 1 through to phase 4, you look at the success rates of trials that include a biomarker of some sort, and you look at those that do not. Consistently, there's a large difference in the efficacy rate of those that include some form of marker. So the Herceptin example Richard gave, I think, is a great one, and I think that sort of post hoc analysis is extremely valuable and shows that we can do it. I think we just have to do it more.
And following up on that, Eoin, and I think also especially on the points made by Richard in terms of integrating this subgrouping of patients. I think we're all tired from COVID research and COVID pandemic, but one of the good things that came out of this to me is actually the remap approach to clinical trials being approved by and actually encouraged by the WHO. The remap is a platform trial where you allow testing different interventions for different domains, meaning that patients can actually, if they fulfill the inclusion criteria, participate in these different interventions and be included in this way to test more interventions in a randomized setting than just one in one clinical trial. What I miss from this perspective is a systematic way to adapt which patients would actually be allocated to which subgroup. And I strongly believe that building on prior clinical trial data and on prior real-world data, on laboratory knowledge, we can integrate that into platform trials, essentially turning the full population into patients having the option to go for what we would consider standard of care at a given time point, or to go for the randomization based on their subgrouping. And I just found a prostate cancer trial on clinicaltrials.gov, which actually allocate patients based on genetic scoring, assessed by machine learning, to standard of care or a number of different interventions guided by this genetic score. And this is really what I believe we should aim for, but doing it in a platform to allow us to build in real time on top of the prior results.
Thank you very much, Eoin and Carsten. So my next question is, are adaptive trials useful? And if yes, when? And what type of adaptation you think would be most useful? And I have this question for, Richard first, and then Eoin.
So to kick off, I mean, my personal view is yes, they are useful, and I'll speak in a few minutes to areas where they might be used. The reality, at least in pharma, is that they're not used that widely. And there are undoubtedly challenges in delivering adaptive trials. I think some of that is driven by pharma's desire to do what the regulators want, because ultimately, if we don't persuade a regulator, we don't get any drugs approved. And adaptive trials may not be the best thing to do for the pivotal phase 3 type trials. But we tend to get pharma tends to get a bit scared of them in earlier stages as well. However, having said that, personally, I do find them extremely useful as a means of beginning to approach some of the problems that Carsten and Eoin in particular have just been talking about. Maybe I can use an example to again build on what Carsten was saying, and this is a breast cancer trial, which may be known to some of you, which is the I-SPY 2, I think it is now, trial, where it's another of these platform trials where patients are adaptively allocated to different drugs. You can rotate drugs in, you can rotate drugs out, and the randomization to which drug you receive adapts depending upon which is looking to be the most promising drug of the ones under study, so that the most promising drugs get studied quickly. But what I find particularly interesting about that trial is it also incorporates, I think it's 11 or 12 different biomarkers of the type of breast cancer that the patient might have. And so you not only begin to adapt a population to a drug, but you begin to understand and explore which biomarkers actually seem to be the most predictive of response. So I think these kind of trials could potentially be an extremely interesting way of starting to tackle this problem of which patients should we be treating, which subgroups should we be treating. And I hope that they can also, to some extent, be informed by, within the trial, by more advanced methods for beginning to work out what might be the predictive biomarkers when you might not have actually predefined them at the beginning of the trial. I-SPY 2, everything's predefined. You know, there are these 10 or 11 biomarkers. There are no new ones brought in, at least not from within the trial. But why wouldn't that be possible to do in some future state? So it's one example of adaptive. I won't go to more. I'll stop and let Eoin take over.
Thanks, Richard. I really like the idea of adaptive trials, in part because they just make a lot more sense than a conventional trial. You don't need to wait to the end to get a binary result, whether a single intervention has made your pre-specified endpoint. That just doesn't feel smart enough. Being able to understand what you're doing as you're doing it has to be a conceptually better approach. So conceptually, I think they're a great idea. Clearly, they are most in use at the moment, where we're trying to do so-called seamless adaptive designs, where you're going straight from phase 3 into phase 3, which perhaps isn't such a big difference, because phase 2 and phase 3 being a bit more similar in the first place. Perhaps they got more to offer at an earlier stage, where we can learn how to tailor an intervention to the appropriate population. I think one big challenge for adaptive designs is the regulatory approval, because beginning a trial without being completely confident that you know the result, if the result has not been pre-specified, that you're going to learn it as you go along, knowing that that's going to be acceptable for regulatory approval downstream, I think has to be demonstrated. I think before people will buy into doing it in the first place. But conceptually, yes, they're as a design, they'd be great. For me, perhaps their success hinges most on an ability to identify in an a priori way the right surrogate markers and the right surrogate endpoints to base the adaptation on. I would be nervous about learning those as you go, because for me, the risk of overfitting to a small or even growing population is quite strong. But if you can have those defined up front, and obviously ML approaches are ideally placed to define those surrogates, and from much larger data sets, then an adaptive design, once you're confident in those markers, becomes absolutely sensible. And from a biological mechanistic point of view as well, if I'm confident in my surrogate, let's concentrate on the surrogate and adapt dose, adapt patients as we go.
Wonderful. Thank you so much, Richard and Eoin. Then I have a follow-up question. Can we deal with disease-specific challenges to run more effective trials? And can we improve outcomes for patients while doing so? Pierre, and then Carsten.
Well, as we said, there is prior knowledge about the disease and the different forms of the disease. We don't always have biomarkers. We have heard a lot about biomarkers. But sometimes you don't have biological markers, but you may have statistical or artificial intelligence scores or types of patients that may play or may be used as or play the role of biomarkers, and that would also be very useful in the sense that first, a new drug would be better approved or better received for non-responders to previous drugs. And that's the population in which it has the best probability of being superior to the comparator. And also another point which is very important, and I said that before, the risk of toxicity or adverse events. The risk altogether is very important to figure out and measure quantitatively. And this this is something that can probably be prepared before the clinical trial. How am I going to measure the risk? How am I going to integrate different types of risks? And how am I going to come up with a definitive score of risk. And I believe that this risk score is very important and can solve many problems, and that would really make a difference for some drugs. A drug might not be more efficient than the comparator, but if it really comes with lower toxicity or risk, and the risk might be in the long term, then it would be superior to the comparator.
Building on that, I think it's a very important perspective that we should model and aim to model both the adverse events or risk for the patients and the benefits for the patients upon a given treatment. And as an example of this, in the CLL-CLU collaboration, an EU-funded collaboration led by Sigrid Skånland, we aim for in CLL, chronic lymphocytic leukemia, to combine data markers and biomarkers. And that's really an area where I want to put your interest, because we see multiple clinical trials with a joint biomarker programs, translational studies, and focusing on one omics approach or another omics approach, or maybe two of them, but often not including the clinical data, not including the adverse event data into this modeling. And we see that the commercial pharmaceutical companies publish on one or the other of these markers, but they do not provide these data for modeling afterwards. And I think if we can combine these data, both the clinical databases and the biomarker and translational studies, and use them for modeling across different trials, we'll be able to actually combine data and biomarkers into subgroups with specific risks. And this is really what we aim for in this CLL-2 collaboration that I hope we can extrapolate to a number of different disease areas.
Thank you so much, Pierre and Carsten. Andreas, I guess it's time for our next question. Can we improve clinical trials by using real-world data? And of course, some of you already have alluded to this in your previous comments, but I'd like to really ask you to answer this question yet again. How do you see this real-world data improving clinical trials? And I have again, Carsten, Pierre, and Richard.
Maybe I can just follow up. So emphasizing work of your group, Mihaela, on synthetic twins. One of our major problems is that if we look at our patient populations, colleagues of mine have demonstrated for multiple myeloma, as an example, that only half of the Danish patients would actually fit into any clinical trial. Thus, for half of our patients, we need to extrapolate from clinical trial data. And we do that more or less blindly. We don't know if the toxicities or the efficacy for these patients not fulfilling the inclusion and exclusion criteria would actually indicate that they would benefit from a given treatment. The synthetic twin, even though I would still challenge you to further develop it, gives the chance to use the real-world data as more or less a randomized clinical trial by identifying the outcome for a similar patient as the one I have in front of me in the clinic. And I think this could really help us by using real-world data to extrapolate in a smarter way the findings from clinical trials and help us to provide a better prediction, a better idea what would be the right treatment for a specific patient in front of us. So essentially taking what I would consider the art of being a medical doctor or physician to actually a data-driven or evidence-based art of being a physician.
If I can just elaborate on this, when you perform a clinical trial, you just draw one ball from an urn of balls, and it's significant or it's not. But you would like to replay it several times. You would like to try again and again and again, and finally come up with a real mean or average effect. And so the question would be, could we replay clinical trials by replacing the participants by people from the real world population that might look like them, but are not exactly them? their nearest neighbors or people in the middle of, or, you know, at equidistance from two different patients from the trial? And could we figure out what would be the result? So in other ways, can we do some kind of extrapolation, bootstrapping of the clinical trials and come up with a p-value and a quantity of effect with an uncertainty, with a 95% confidence interval, for instance? That would be very useful.
And if I can just have the comment of bringing a few things together, for me, one of the biggest opportunities of real-world data is to tackle this problem of disease heterogeneity. It's been mentioned, I think, a few times in different ways. Let's just really make it very explicit. When we think about it, most diseases are diagnosed today using 19th and early 20th century technology. You know, if we were actually started, what is the patient complaining of, and what signs, what clinical signs do they have, and maybe what are the changes on some very basic investigations? You know, that's all been, that technology has been available 100 years or more. If we were going about diagnosing diseases today, we might include those, but we'd include a lot of other things as well, and we'd come up with an awful lot more specific diagnoses. And I think that's one of the key areas of opportunity for me. That's pattern recognition, which computers are very good at, from complex patterns, but allows us to go into much more granularity about the patterns than we were capable of doing 100 years ago.
I would just add one thing to that, Richard, which I completely agree with all of it. That heterogeneity is only the tip of the iceberg, because the minute you add in multimorbidity,
yep,
then all of a sudden it's not just this heterogeneous condition, but it's this one overlapping with this one, overlapping
yep,
with this one. So actually there are as many diseases as there are patients, really, and that just isn't captured. We try and make trials as homogeneous as possible.
Yep, it's a great point.
Thank you all very much. The last question, Andreas, is, can we transform post-marketing evaluation of benefit-risk balance and assess efficacy? And this question is again, Pierre and Richard.
Can I borrow something to start with, just that Richard said earlier, which I thought was a great example of this? The example you gave of IL-6 in COVID, which just shows you that the way we treat patients is dynamic. So the clinical trial result you get in your trial in 2022 may not be the result that you expect to get in 2032, because other treatments for other coexisting conditions will have changed, and the context of medicine will have changed. So there is scope, again, and this can use an estimation of individual effects in real world data, for example, to effectively monitor the impact in the way that we do in a very clumsy way with post-market surveillance. Post-market surveillance is there to make sure you're not killing anyone with your treatment and not having untoward adverse effects, but we could really estimate the ongoing efficacy and how that changes over time by using real-world data as well. And that feeds forward in letting us know whether our treatments are still efficacious or whether they've become more efficacious and who we should use them in. But it also can feed backwards into the next round of trial design. So I think there's huge scope for using that data, and it's only very recently that it's becoming systematically collected in a way that makes it usable. Not easily usable, and I think part of the restrictions on regulation and access are part of a discussion of a challenge for that. But with federated data access approaches, swarm learning methods, those are ways in which those data types can perhaps become more accessible, more useful in the future.
Maybe if I can add, there are ways to try and correct for differences in target population, between target populations and real world populations, but these corrections are very suboptimal because they only correct for a few characteristics, personal characteristics that seem to differentiate the population enrolled in phase 3 trials and the population that actually received the drug. So here again, the concept of digital twin or non-event, fictitious non-events would be important. So what would happen to the patient that received the drug if they did not receive the drug? And what would happen to patients who did not receive the drug if they had received the drug? So This would be the way of actually measuring the actual drug effect in the population. And when I say drug effect, again, I want to emphasize that it's a benefit-risk balance, because patients can be harmed by drugs, as you all know, and can be hospitalized, and some can even die because of drugs, drug interactions, drug overdoses, whatever. But they can die due to our medical interventions.
Maybe I can come back to dose briefly, because I said earlier that I think it's still something that is a problem in understanding what's the best dose of a drug, even at the population level, even for average patients. It's probably not something that's been done very well for many drugs up till now. There is a big opportunity to understand dose post-approval and how that might vary in different populations, because that's, as we've already heard, that's the point at which the drug actually gets expanded out into a much broader patient population and into phenotypes who would not have been eligible for the trials in the first place. So one of the big opportunities for me post-approval is to actually get into the question of what dose should we be giving? And that may be one of the more effective ways of persuading pharma that we need to do a better job of understanding dose even pre-approval.
Thank you so very much, Carsten and Pierre and Richard, for all the important insights and lots of food for thought that hopefully we can synthesize and bring forward to this community next time. In view of time, I think we have time for 2 questions from the audience, and I think some questions have already been asked. I think the first question was from Rahul. Am I correct? Rahul, would you mind asking your question and maybe providing your answer?
Thank you. Can you hear me?
Yes.
Thank you very much. Yeah, I was just wondering about any limitations with using the synthetic data, or will there be any bias from the underlying data set on which it is generated, especially when we are trying to use it on clinical trial? And if so, which phase of the trial will it be more effective? Thank you.
So I'd given you a response of sorts in the chat about this, but effectively, I mean, I think there are some examples that synthetic data can be great, and I think it has huge potential. It does depend on how it's synthesized. So how have you learned the disease characteristics to produce your synthetic data? And that perhaps is the key consideration. If you can generate a synthetic data set that is appropriate for the intervention you're going to model, then I think you can do a lot better in terms of estimating what likely effect size you're going to have. If you can do something analogous for your intervention, then you can put the two together and you could have a very powerful way of improving your study design. So yeah, I think synthetic data, yes, but depends on where it comes from, where it learns from. Stage of trial design, where it could operate best. I think it has most, if you remember at the beginning, my little sketch where you have increasing data as you go through the trial phases. I think the further you go through that process all the way out to post-market use, then the further you go, the more actual data you have. So perhaps synthetic data has most to offer at the earliest stages. That's quite a simplistic interpretation, but I would expect that you have more actual data to model as you go through, and therefore probably the earlier, the more informative.
Just to add, I would totally agree with that. I mean, particularly in the later stages, if you want to persuade a regulator or for an approved drug, if you want to persuade an expert body, they're going to need to see data from real patients. The most useful thing is to inform who are the right real patients to study and how much should we give them and how do those two interact. So I think you're spot on that the biggest utility will be in the early stages in helping us set the trials up in the right path.
Can I ask you one thing, Richard? This is off script. Do you think there is a tension between stratification of a heterogeneous population and a commercial desire to treat the whole population? So if I tell you that you've got a heterogeneous group of patients and you've got either the choice of stratifying it to treat half of them bet, half of them in a targeted way that you think will be more efficacious, or you can chance your arm and treat the lot of them where your subsequent trial may suggest that actually that treatment applies to the whole population and you've doubled your market. Is that a tension within pharma? I guess that it must be.
I think it is still a tension, yes. There's still the belief amongst many people, and I don't think it's unique to pharma. I've seen it in many clinical academic grant proposals as well. There's still a belief that the drug will work in everyone, or the treatment will work in everyone. And so that tends to be the model that people go for. Plus the fact that we talked about many times that we may not know where else to look. I think as soon as you know where else to look, actually there's no problem going and looking in a relatively small population. You know, we've had the example of, well, we know in the cancer area in particular, there are now quite a rapidly growing number of molecules specifically targeted to relatively small subsets. And pharma companies are going after that because they can identify the subset. And so that's not such an issue. And then there's also the famous example of abacavir, where being able to identify the subset who should not get the drug actually turned it into an extremely commercially successful drug, whereas previously it hadn't been, because there was a rare but often fatal side effect in a small percentage of patients, which meant it was left to use only as a last resort. So I think the tension is still more a cultural, behavioral one, often of the investigators and not so much of the pharma company. But it's the best solution is get more granular at understanding who are the patients we should actually be treating.
And maybe I may pick up on that because I saw from Burlis the question about how is the position of the regulators towards this. And I think the regulators also, based on the new EU-based directive on medical devices, try to be very forefront thinking in terms of allowing these kinds of algorithms and a combination of data-driven and biomarker-driven trials to be allowed. But they put the same restrictions and requirements in terms of evidence as we have seen for randomized clinical trials, meaning that it will be a very slow process and it's very expensive for us as academic researchers and for companies as pharma to develop this. Thus, we need to join forces between pharma, academia, and regulatory bodies to define a middle way or balance to actually make this feasible. Because when I discuss these trial designs with pharma companies, they're often very scared by this medical device approval.
Thank you very much for all the insights and for the off-script discussion, which I thought was very interesting. In view of time, I just want to thank again to the four of you for this very fruitful discussion and food for thought. And I guess it's time now for us to get back to work to try to understand whether machine learning can address some of these issues or not. Andreas.
Yeah, also for me, thank you very much to all of you. Thank you very much for the questions from the audience as well. Thankfully, we could ask Burley's question in the chat as well. Coming up next, so 7th of July, we will have the second part of this topic in a session on how machine learning can transform clinical trials. And we already dived in quite deep today, and I'm sure we will pick up in July as well. This session you can watch and share in a couple of days when it comes up on YouTube. And if you have any other colleagues, practicing clinicians who would benefit from these sessions or would like to let their voices heard as well, please go to our website at the Schaar Lab and subscribe to this letter or send them the link so that they can subscribe because you already are. And thank you very much for coming. And I hope I will see you in July.